white-eye gene from their mother's X chromosome and no-eye color gene from their father's Y chromosome. The outcome of Morgan's Punnett square is consistent with the actual breeding outcomes. The Morgan hypothesis isn't just about predicting eye color in fruit flies. It is also relevant in explaining the inheritance of a number of genetic diseases in humans. Genes that are located on one sex chromosome but not the other are referred to as sex-linked. Nearly all sex-linked genes are found on the X chromosome and are often referred to as X-linked genes. Humans carry genes related to color blindness, hemophilia, and muscular dystrophy exclusively on the X chromosome. The fact that males have only one copy of the genes for these traits puts them at a much greater risk for these illnesses than females, as they have no chance of inheriting a second good gene that will mask the expression of a bad gene they may inherit. Sex chromosomes are a special case. All the rest of an individual organism's chromosomes, called autosomes, match up very well with the chromosome with which they are paired. Since Sutton and Morgan, it has been well established that each chromosome has a large number of genes on it, some with identical alleles to the chromosome with which it is paired, and in other cases, different alleles. As we know, Mendel hypothesized that genes sort independently. But how can genes on the same chromosome sort independently if it is chromosomes rather than genes that assort independently during meiosis? The answer is that Mendel was partially correct. Genes that are on different chromosomes do always assort independently, and the genes for most of the traits Mendel studied were on different chromosomes. But it would appear that genes on the same chromosome would always be linked or inherited together. There is, however, a phenomenon, crossing over, that you may remember from your study of meiosis that prevents all the genes on a chromosome from always being linked together. During prophase 1 of meiosis, homologous chromosomes intertwine. The points at which they cross over one another form chiasmata. At chiasmata, chromosomes may exchange sections with one another in a process called crossing over. If one gene is located on one side of a chiasmata and another on the other side, after crossing over, the two genes are no longer linked. If two genes are on the same side of the chiasmata after crossover, they remain linked. Obviously, the closer two genes are to one another on a chromosome, the more likely they are to be linked, or in other words, to be inherited together. There is almost always at least one or two crossovers per pair of homologous chromosomes during meiosis. If genes are located far apart on a chromosome, they are likely to be frequently separated during crossing over and act almost as if they were independent of one another, just as genes on separate chromosomes do. However, if two genes are very close together on a chromosome, say right next to each other on the end of a chromosome, they will almost never be separated from one another during the process of crossover and thus almost always be inherited together. In light of this information, Mendel's law of independent assortment should be modified to say genes located on different chromosomes always assort independently during meiosis. As a result of crossing over, genes on the same chromosome may or may not tend to sort independently, depending on how far apart the two genes are on the chromosome. The further apart they are, the more likely they are to sort independently. There are also a number of other variations regarding inheritance that Mendel's work didn't deal with. Incomplete dominance, multiple alleles, and codominance, polygenic inheritance, and environmental effects on gene expression. Mendel ran into a rather straightforward situation with the pea plants he studied. The genes for all the traits he studied were either or, completely dominant or recessive. Probably a good thing, as it made the argument against blending stronger. But interestingly, there are occasions in nature when something very much like blending appears to occur. For example, when homozygous red-flowered snapdragons are crossed with homozygous white-flowered snapdragons, the flowers of the F1 generation aren't all red or all white. Because the alleles for neither flower color is dominant, the flowers of the now heterozygous plants are pink. This pattern of inheritance is called incomplete dominance. The reason we say that blending only appears to have occurred is that in the F2 generation, both red flowers and white flowers return alongside the pink flowers of the remaining heterozygous plants. If real blending had occurred, there would be no red or white, as they would never form again, as the genes for red and white flowers would have blended to form pink flower genes. Mendel was not only lucky because the traits he had to deal with were all clearly dominant or recessive, but also because none of the traits he experimented with ever appeared in more than 2 different forms. But more than 2 alleles are common for a large number of genes in nearly all species. However, any individual can only have 2. A common example of multiple alleles in humans occurs in blood type. 3 different alleles are responsible for the formation of 4 different types of human blood: A, B, AB, and O. The 3 alleles are usually designated IA, IB, and I. These 3 alleles are responsible for directing the synthesis of the glycoprotein identification markers that protrude from the surface of red blood cells. IA and IB control the synthesis of the glycoproteins A and B, respectively. The I alleles produce no glycoproteins at all. These 3 alleles produce one of 6 different genotypes in a human being: IA, IA, IB, IB, IA, IB, IAI, IBI, or II. Both the IA and IB alleles are dominant to the I allele, and thus form A glycoprotein and B glycoprotein, respectively, if in a heterozygous pairing with an I allele. However, if IA and IB alleles are paired, both A glycoprotein and B glycoprotein protrude from the surface of an individual's blood cells. Since both A and B glycoproteins are present in approximately equal amounts and maintain their own unique characteristics, they are said to be codominant to one another, while the snapdragon example is considered incomplete dominance because the traits of red and white are no longer discernible as individual colors. Being IA, IB, and thus having AB blood makes one a universal blood recipient. For obvious reasons, the body of an individual with AB blood wouldn't make A glycoprotein or B glycoprotein antibodies. That would cause type AB or AB to clump up and form clots. Not having those antibodies allows AB individuals to receive blood from A, B, AB, and type O individuals, as type O blood has no glycoproteins to be attacked by antibodies. Since they have no glycoproteins to be attacked by antibodies, O individuals are known as universal donors. But since they have both A and B antibodies, type O individuals can only receive blood from other type O individuals. Mendel's work also didn't look at the traits that are controlled by more than one gene, of which there are many. 2 examples in humans are eye color and the darkness of one's skin. Eye color is controlled by at least 2 and skin color by at least 3 separate, incompletely dominant genes. The greater the number of genes that control a trait, the more subtle the variations in a trait. If the alleles are incompletely dominant, as they are in the case of eye and skin color, that increases the number of variations further. The genes that control eye and skin color do so by controlling the amount of the yellowish-brown pigment melanin produced on the front of the iris or in the skin. An iris with no melanin in it would naturally be blue, reflecting blue light just as deep bodies of water do. The eyes of babies whose eyes will eventually become darker often start out blue because they haven't yet started producing melanin. If we presume that eye color is controlled by two genes with two incompletely dominant alleles, then individuals can potentially have 5 different eye colors, depending on the number of melanin-producing alleles the individual inherits from their parents. An individual that inherited no melanin-producing alleles would have light blue eyes. An individual with 1 melanin allele, deep blue or green eyes. An individual with 2 melanin alleles, deep green or light brown. An individual with 3 melanin alleles, medium brown. And an individual with 4 melanin alleles would have dark brown eyes. Variations in skin color occur in a like manner, except that 3 or more genes create 7 or more shades of skin color. But environmental factors, like exposure to the sun, also affect skin color. Not only skin color, but the expression of all traits, weight, height, personality, intelligence, athletic and musical abilities, etc., are influenced to a greater or lesser degree by environmental factors. However, we humans are unique in that we can control our environment and thus the expression of our genes to a much larger extent than other living organisms. Thus, while the genes we inherit from our parents certainly influence who and what we are, they do not define our destiny. Though scientists certainly don't know exactly how much complex traits, such as intelligence and musical ability, are influenced by our genes, and how much by our environment, they certainly know that their development and expression can only be maximized through hard work, determination, and exposure to an environment conducive to their development.